Hello, my name is Dr. Nichola Conlon, and I am the founder and the lead scientist at Nuchido Laboratories. Now, when I founded Nuchido, it was my mission to translate the latest science in aging research into consumer products that actually work. I wanted to bring out good products that have good science and good testing to support them. And for that reason, we did a clinical trial on our NAD boosting supplement, Nuchido TIME Plus, to prove that it actually works. Because what many people don't realize is that the vast majority of supplements out there actually don't have any clinical testing because it's not actually a legal requirement. So many companies simply don't bother. So at Nuchido, we invested in doing a gold standard, placebo-controlled, double-blinded, randomized crossover study. And in this webinar, I am going to be explaining the background behind our product and how it's been designed and the clinical results that we have that will be going on to be published in a peer-reviewed scientific journal. So to begin, as a brief overview, Nuchido Time Plus is a dietary supplement. It's designed to boost levels of an important cellular molecule called NAD. And we refer to it as a second generation NAD booster because it works in a different way and a far more superior way to previous NAD boosting supplements that are out there, such as NR or NMN. But first of all, what is NAD and why is it important? Well, NAD stands for nicotinamide adenine dinucleotide, first of all, but that's a bit of a mouthful. So everyone just refers to it as NAD or NAD+. And NAD is a natural molecule. It's found in every single cell in our bodies, and it's actually involved in hundreds of different cellular reactions. But perhaps it's most famous for two main things. The first is cellular energy production, where it helps the process that converts the food that you eat into the energy that all of your cells need to survive. And secondly, it's critically important for switching on cellular maintenance and repair processes. So as a general rule of thumb, if you have high levels of NAD, then you get lots of energy production and repair. And if you have low NAD, then you get low levels of energy production and repair gets turned down. And one of the main reasons that NAD has the ability to switch on and off all of these maintenance and repair processes is because NAD controls the activity of a family of proteins known as the sirtuins. Now, sirtuins are often called the longevity proteins because when they are activated, they in turn switch on a whole host of downstream processes that promote the health and the longevity of the cells. And there have been lots of studies to show that activation of the sirtuins results in extending the healthy lifespan of cells. But the key thing here is that they are absolutely dependent on NAD to work. Without NAD, they could not perform any of their critical roles. So why is NAD actually linked to aging? Well, the problem is that NAD levels have been found to decline with age. So when you are young, you have really high levels of NAD, but as you get older, they decline. In fact, cellular NAD levels are found to decline by about 50% every 20 years, which is quite a lot when you think of how important it is to the body. And when NAD becomes persistently low, both energy production and repair are turned down, resulting in an accumulation of damage, increased biological age, and many of the symptoms and negative health consequences associated with aging. So scientists said, okay, so we've got this molecule, it's obviously really important, and it declines with age, and this causes quite a few issues. So what happens if we just don't let it decline and we keep levels high as we get older, or indeed we try and restore NAD back to youthful levels? So this is exactly what they have tested, and they found that restoring cellular NAD levels resulted in incredible benefits to cellular health. And as a result, many scientists believe that maintaining levels of NAD as you age is one of the most promising ways to combat the aging process and maintain your health span, which is the proportion of your life that you will live in good health. So a key question is, how would you boost NAD? Well, first of all, it should be noted that you can't just take pure NAD orally. This is because NAD is an unstable molecule that degrades very easily, so you can't just put it in capsules. And if you did, and it survived this process, it would almost certainly be broken down in the gut. The other thing with pure NAD is that it's quite a large molecule, so it can't freely diffuse across the membrane of many cells. Because of these issues, one method that has become a popular way to boost NAD is to use supplements that contain the building blocks that the body uses to make NAD. These are known as precursors, and common ones are NR or NMN. These are basically the raw materials that the cell uses to make NAD, with the idea being that if we give the body more of the raw material, then hopefully it will use it to make more NAD. But unfortunately, it is now known that this method does not address the root issues that are causing the NAD to decline in the first place. And there is now also evidence that using this approach may cause further issues such as methyl donor depletion and inflammation, which I will explain shortly. So how do we boost NAD efficiently? Well, to date, all the latest research shows that the best way to boost cellular NAD levels is to, surprise, surprise, fix the root causes that are leading to NAD decline in the first place, which unfortunately supplements like NR and NMN ignore. And if you're interested in learning more about the scientific detail on this, you should read this paper that I published recently, which is open access and free to download. So what causes NAD to decline? Well, there are known to be two main issues. The first is the main enzyme in the cellular pathway that makes and recycles the majority of your NAD actually declines with age. This enzyme is called NAMPT, and it's an enzyme found in the salvage pathway. The second reason is that the demand for NAD actually increases as we get older, and this is mainly due to chronic low-grade inflammation. And together, this reduced NAD production and increased NAD consumption effectively creates the perfect storm for NAD to become depleted. And just to note, there is actually no evidence that NAD declines due to a shortage of NAD precursors such as NR or NMN. So, to appreciate why these issues cause NAD decline, it's good to understand a little more about how our cells usually make NAD when they are young and healthy, because our body is actually pretty good at making its own NAD. So, in young healthy cells, the majority of NAD is made by the salvage pathway. And at the bottom, you can see that as NAD is used up by cellular processes, such as the DNA repair enzymes and the sirtuins, it is broken down into a precursor that is called nicotinamide. And this is abbreviated to NAM in these diagrams. Now, in healthy young cells, this nicotinamide is simply recycled straight back into fresh NAD again via the salvage pathway. And this means that as NAD is used up by the cells, it's automatically restored without any need for any external input. And this makes sense, really, because why would our cells want to rely on raw materials from the outside to produce such a critically important molecule? They would want to have their own fail-safe way of recycling NAD breakdown products back into fresh NAD again. So what goes wrong with age? Well, we now know that in older cells, the salvage pathway becomes dysfunctional, meaning that less NAD can be made and recycled. This is because a key enzyme in this pathway called NAMPT, which can be seen at the top, declines with age. And this is a problem because NAMPT is what we call a rate-limiting enzyme, which means that the amount of NAD produced is directly dependent on the amount of this enzyme. So when levels of NAMPT decline with age, this means as NAD is used up by cellular processes, it's broken down into nicotinamide, but this nicotinamide can no longer be recycled back into NAD again, and this causes NAD levels to ultimately decline. This reduced capacity of the salvage pathway to recycle nicotinamide also causes further problems such as methyl donor depletion. This is because as NAD is being used up and broken down into nicotinamide, excess nicotinamide starts to accumulate in the cell because it can't be recycled. And cells don't like it when things start to build up. So to compensate, cells start producing another enzyme called NNMT, seen here on the left. And what this enzyme does is it adds a methyl group onto the nicotinamide, signaling for it to be removed from the cell as a compound called methyl nicotinamide, or MENAM for short. This is a problem because the cell has to use valuable methyl groups for this process, rather than using them for more important processes like DNA synthesis and epigenetics. Now, aside from reduced production and recycling of NAD, the other main reason that NAD declines in older cells is due to chronic low-grade inflammation, which leads to increased NAD demand and wastage. This is predominantly due to the overexpression of an inflammatory protein called CD38, which can be seen at the bottom here. CD38 is an inflammatory enzyme that uses huge amounts of NAD. To put this into perspective, for every 1 cycle of the CD38 enzyme, it uses around 100 molecules of NAD. And just like any other process that uses NAD, it breaks it down into nicotinamide. So this results in even more nicotinamide accumulating in the cell, and this leads to even more methylation and excretion, making these problems worse. So with all that in mind, what's the problem with precursors? So what happens when you take a precursor to try and boost your NAD levels? Well, first of all, you can clearly see that precursors such as NR or NMN do not address any of the issues that are causing NAD decline in the first place. But more worryingly is that by ignoring these issues, precursors may actually be inadvertently causing other issues. For example, take inflammation. We know that the inflammatory protein CD38 has a higher affinity for NAD than any of the more beneficial pathways, such as the sirtuins or the DNA repair enzymes. And this basically means that if NAD is available in the cell, then CD38 will grab it before any of these more beneficial processes can get hold of it. So if CD38 is not inhibited before boosting NAD by any means, then NAD may inadvertently be driving inflammation via CD38. Also, think about what happens if you're boosting NAD, but the salvage pathway is not working functionally. Because basically, once this NAD has been used once and then degraded into nicotinamide, it will start to build up in the cells if it can't be recycled, which means it needs to be methylated and excreted, resulting in even more methyl donor depletion. And actually, if you look at some of the clinical trials that have been using the precursor NR, you can see that as the dose of NR is increased, urinary excretion of methyl nicotinamide also increases as the participants' bodies struggle to deal with all the excess nicotinamide, which is why many people who are supplementing with NR or NMN end up having to also take a methyl donor supplement like trimethylglycine to counteract these negative effects. So when we were designing the Nuchido TIME Plus formulation, we wanted to take a completely different approach and actually fix the root causes of NAD decline rather than ignoring them. We wanted to design a product that would switch back on natural NAD production and recycling, that would inhibit inflammatory processes that waste NAD, and also prevent this methylation problem. So this is exactly what we did, and now I'm going to take you through how all the ingredients in Nuchido TIME Plus work to fix these issues. So one of the first things we wanted to do was to use ingredients that switch back on the NAMPT recycling enzyme. So in our formulation, we included Sophora javanica, which is an extract that contains the active ingredient quercetin, which is known to be a direct activator of the NAMPT enzyme. And because the NAMPT enzyme is just so important for NAD production, we also included another ingredient in our formulation called alpha lipoic acid, or ALA for short. And this also activates NAMPT, but via another pathway called the AMPK pathway. Now, this approach, that means that we're activating this important enzyme by not just one, but two different mechanisms. In the cell, NAD is also known to flip between two different forms, which can be seen here at the bottom. One is NAD plus and one is NADH. And as we get older, the ratio of NAD plus to NADH shifts more towards having more NADH. And when it comes to healthy aging, it is important to push it back in the more youthful direction of having more NAD plus. Now, alpha lipoic acid is known to activate another enzyme, which means that it forms a dual purpose in our formulation. And this enzyme is called NQO1, which is known to convert NADH back into NAD+, pushing it back in a more favorable, youthful direction. Another ingredient that we included in the design of this formulation is green tea leaf extract, which contains a powerful active ingredient called EGCG. Now, EGCG works to inhibit this methylation enzyme NNMT. And what this does is it promotes the recycling of nicotinamide back into NAD rather than it being methylated and excreted. This therefore avoids the depletion of methyl groups that are associated with the other NAD boosting methods that are out there. The Nuchido TIME Plus formulation also includes an unusual ingredient, which is parsley leaf extract. The reason that we use parsley leaf extract is it's included because it contains high levels of an active ingredient called apigenin. And apigenin is known to be a CD38 inhibitor. And it's known that just by inhibiting CD38 even just a little bit, you can boost NAD levels because remember, CD38 is such a prolific waster of NAD. Also, by inhibiting CD38 and not ignoring this issue, it ensures that any new NAD that is actually produced is being used for beneficial processes like the sirtuins and DNA repair and is not grabbed and used up by CD38 to inadvertently drive inflammation. And finally, we do also include a precursor in our formulation, but the precursor that we use is nicotinamide. We do not use the precursors NR or NMN because they are not freely diffusible across cell membranes. They actually require special channels to cross into cells. Therefore, they cannot access every cell type. Only cells that have the correct channels, they can get in. Whereas nicotinamide is actually freely diffusible across all cell membranes, and we know that it is the preferred precursor for the cell to use to make NAD. We also include some other supportive ingredients, including vitamin C, zinc, and also black pepper, which contains the active ingredient piperine, which helps to aid absorption and improve bioavailability. So now the clinical trial. And first of all, I'm going to start by taking you through how the trial was designed. So we performed a placebo-controlled, double-blinded, randomized crossover study, which seems like quite a mouthful, but let me explain what that all means. First of all, placebo-controlled means that as well as taking the real supplement, participants also took an identical-looking placebo capsule. Double-blinded means that neither the participants nor any of the researchers involved in the trial knew which were the real capsules and which were the placebo capsules. They were simply labeled A or B for the duration of the trial and its analysis. Randomized crossover means that the participants took both the placebo and the Nuchido TIME capsules. To do this, the 28 participants were divided into two groups randomly. One group received the placebo capsules first, followed by a washout period, and then the Nuchido TIME Plus capsules, whilst the other group started with the Nuchido TIME Plus capsules, then had a washout, and then took placebo capsules. Now, this type of trial design is much better than a non-crossover study where one group takes a placebo and the other group takes the real thing. Because in this design, you are comparing two completely different groups of people. Whereas with a crossover design like this, everyone is directly comparable to themselves. In our trial, we used 28 people. They were aged from 20 to 80 years old, and they were both male and female. which is unusual because many trials for NAD are only done in males aged around 50 to 60, which I'm sure you would agree is not reflective of the typical population. So the results, what did we actually find in this trial? Well, the first obvious thing to measure was NAD levels. And it was found that after 28 days supplementation with Nuchido TIME Plus, NAD levels were significantly increased in the participants compared to when they were taking the placebo capsules. During the clinical trial, we also measured NAD at weekly intervals, and Nuchido TIME Plus was found to significantly increase NAD levels after only 7 days supplementation. Now, one question that I get asked a lot is if we can measure NAD in a lab, then can you get an at-home blood test to measure your own NAD levels? Well, unfortunately, the answer is no. This is because NAD is a very unstable molecule, meaning that it rapidly breaks down outside of the body. So unfortunately, you can't just put a blood sample in the post and send it to a lab, as it would be completely degraded after about an hour. In our clinical trial, when the blood samples were taken by the lab that performed the trial, they were immediately preserved to prevent this from happening. The next thing we wanted to find out was our formulation actually fixing one of the main root causes of NAD decline and switching back on that all-important NAMPT enzyme? And yes, I'm pleased to say it does. So Nuchido TIME Plus was found to significantly increase the amount of the NAMPT enzyme in the cells of the participants. This means that Nuchido TIME Plus actually addresses the root cause of NAD decline and switches back on your cell's natural NAD production pathway. This is why we call Nuchido TIME Plus a second-generation NAD booster, because it addresses the main root causes of NAD decline, unlike other NAD boosters such as NR or NMN. And Nuchido TIME Plus is the only supplement that has been proven to boost levels of this key NAD production enzyme in a human clinical study. The clinical trial also demonstrated that Nuchido TIME Plus led to increased amounts of the key longevity protein SIRT1 in the cells of the participants. This is significant because, as mentioned earlier, NAD Plus and the sirtuin pathway is considered one of the key pillars of longevity, because together they activate so many other important pathways that promote cellular health. And like NAD, the sirtuins are found in every cell in the body, so increasing sirtuin activity has whole body benefits. Another key finding of the study was the effect that Nuchido TIME Plus had on inflammation. So we know that inflammation is a normal part of a healthy immune system, but we now also know that as we get older, our immune system seems to become chronically overactivated, meaning it produces inflammation even when it isn't needed. This excess inflammation causes damage to cells, tissues, and organs, preventing them from functioning optimally. In fact, we now know that this chronic inflammation is a major driver of the aging process, so much so that scientists now refer to it as inflammaging. Therefore, preventing and reducing chronic inflammation is critical to aging well. In our clinical trial, as well as boosting NAD levels, Nuchido TIME Plus was found to reduce levels of the inflammatory cytokines IL-2, IL-5, and IL-23. Nuchido TIME Plus was found to reduce levels of glycation in the participants. Glycation is an irreversible reaction which sugar molecules get stuck onto proteins within our cells. This causes them to stiffen and change shape, preventing them from functioning correctly. A really classic example of this is when collagen and elastin proteins in the skin become glycated, and this causes the skin to lose its firmness and elasticity and causes wrinkles. And this is why sugar is bad for aging. In our clinical trial, we measured participants' levels of glycated serum proteins and found that Nuchido TIME Plus reduced glycation following 28 days of supplementation. This is because NAD is known to activate a protein recycling pathway called autophagy, which can help remove damaged proteins from the body. Another thing that we were interested in measuring was biological age. Your biological age is the rate at which you are aging on the inside. It's how well your cells are functioning and how much damage they have accumulated. In the trial, biological age was measured using a test called glycanage, which was taken before and after supplementation. Glycanage calculates the biological age by looking at the patterns of sugar molecules on immune cells, which change in a predictable way with age. In our clinical study, Nuchido TIME Plus was found to reduce biological age by an average of 1.26 years after only 28 days of supplementation. And finally, even though all the ingredients in our supplement are known to be safe and well tolerated, some standard liver function tests were also included to further ensure product safety. And it was found that there were no significant differences between the placebo and the Nuchido TIME Plus, indicating no toxic effect on liver function and that the supplement is safe and well tolerated. So, just to summarize, in a randomized, double-blind, placebo-controlled crossover study, Nuchido TIME Plus was found to significantly increase cellular NAD after only 7 days. Unlike NR or NMN, it works by switching back on youthful cellular NAD production by reactivating a key enzyme in the salvage pathway. It was found to increase the longevity protein SIRT1, to reduce levels of the inflammatory cytokines, indicating that NAD is being directed towards beneficial processes rather than inflammatory CD38. It was found to reduce glycation and also reverse biological age. And finally, what do these biomarkers translate to in terms of customer benefits? Because it's one thing saying NAD and NAMPT and SIRT1 increase, but how will this make you feel? Well, as mentioned throughout this talk, we now know that NAD is found in every cell in the body, and it's incredibly important for cellular health. Therefore, it's no surprise that the benefits are body-wide. The top 5 reported customer benefits are increased physical and mental energy. Many customers report a significant reduction in brain fog, particularly in perimenopausal and menopausal women. We see improved recovery after exercise, improved sleep also because NAD is important for regulating your circadian rhythm. And finally, after more longer-term use, we also see a significant improvement to hair, skin, and nails in terms of both growth and quality. Because when your cells are happy, then your body starts investing more energy in the things that it deems less important. So thank you so much for listening to this webinar. And if you have any questions at all, please don't hesitate to contact us. You can email us at care@nuchido.com and we will be more than happy to help.